Hello, everyone, and thank you for joining us for this workshop. I'm going to talk to you about OBIS and how you would go about publishing eDNA data within our database. This presentation has been prepared together with my colleagues, so me, Sarah Suominen, my colleagues Stephen Formel, Elizabeth Lawrence, and Emily Boulanger. And I'm going to give you a short introduction to what is OBIS, how we work with eDNA data, what our data standards look like, and what would it look like in practice to publish data to OBIS. So what is OBIS? OBIS is the Ocean Biodiversity Information System. We are a global open access database for marine biodiversity information. We are part of the UNESCO Intergovernmental Oceanographic Commission. At the moment, we have over 200 million occurrence records from all around the world in over 7,000 data sets. We are also tightly connected with GBIF, so we work together on publishing biodiversity data around the globe. But overall, OBIS is free to use, free to contribute to, and basically what we want to do is try to bring all the information in the world that is collected about marine biodiversity into one place so that it can benefit everybody. And of course, we are not working in one place in the world. We are trying to reach the whole globe, and we are a mature global community that works with a network of 36 nodes. So here you can see a map of where the nodes are situated, and these are national, regional, or thematic nodes. So these are our data providers who actually collect the information from their region or theme and make sure that it is in the right format and publish it on their IPT, which is the Integrated Publishing Toolkit. These nodes span across 99 countries and represent over 1,000 institutions and over 6,000 scientists. So if we look at eDNA data currently in OBIS, as I mentioned, we have 200 million species observations at the moment in around 7,000 datasets. Of these datasets, only 128 at the moment are eDNA datasets. We've only been doing this for a few years, collecting and developing eDNA standards. However, those datasets already hold 44.5 million records. We have 2% of all datasets that are eDNA, but already 25% of all occurrence records in OBIS. So I think this already shows how important this data type will become for our biodiversity information and for the level of understanding that we have on marine biodiversity around the world. So sharing that data is extremely important so that we can actually benefit from all that information that is collected around the world. Now, what is it in it for you? Why would you want to publish eDNA data? Well, why are we interested in eDNA? Obviously, it's a very powerful tool. It can detect species that other methods might miss. It's non-invasive, scalable, as I'm sure you all know. And shared eDNA data is extremely powerful. So it contributes to these global analyses, through us also to UNESCO global analyses that can have policy effects. The data has long-term value, and the sequences themselves become findable via OBIS. So the benefits to you are really in this DOI minting, so you get a citable format of your data set within your publications, and you can also see how many people have used your data sets. It is a long-term archive of FAIR data. We have that global reach through OBIS and with our connections to GBIF. And we can have that sequence search. So you can really start to look at, I have an unknown sequence. Where else is this sequence found, or closely related sequences? Does that tell me something else about my data that I would otherwise have missed? So what's so special? I strongly feel it has enormous potential to expand our access to biodiversity data at scale and capture that different types of eDNA data. And we also have already this great example of great data management strategies that raw sequences are already widely shared in NCBI. So that's a fantastic resource, and it shows us that it is possible to have immense benefit from sharing DNA data widely. However, I believe, and we believe, that NCBI alone is not enough, or other raw sequence repositories, as of course these raw sequences, you cannot really search them in terms of species name, time, or space factors. There's very little links to other metadata like spatial coordinates or sampling data. And of course, you therefore cannot support distribution maps that are linked to the species information that you have. But okay, what's special about the data that we get from eDNA? So OBIS and GBIF both we are used to, and we are built on traditional biodiversity observations, so to say. However, eDNA is quite a different type of data. It is derived information. It's not a direct observation. So there's extensive lab work and bioinformatic processing that is done before you arrive to your species annotation. It also relies on reference databases, which is another one of these decisions that you make, and your results depend heavily on the database completeness. There is still a large fraction of unknown sequences, so we actually cannot give names to those, but we can kind of have these hypotheses of species. And so the Darwin Core DNA extension that I'm going to talk about is meant to record how this work was done and enable future reanalyses and evaluating how well the data is suitable for future work. So eDNA data standards, as you have heard also during this workshop, are a multitude. Well, there's two major eDNA data standards, which is the MIXIS, minimum information about any sequence, usually used within, or often used within NCBI. And then we have Darwin Core, and Darwin Core is the community vocabulary for biodiversity data, which is used within OBIS and GBIF. And what we really do with OBIS and GBIF is that we link that genetic data information or information derived from genetic data to those spatial coordinates in time, and also make links to what we already know from traditional biodiversity observations or existing biodiversity observations. However, in terms of the data standards, it's a rapidly changing field, as you have seen. We have the FAIR eDNA terms that are being developed or have been developed, and also Darwin Core. We currently use something called the Darwin Core archive, but we will soon have the Darwin Core data package, which will also simplify how eDNA data is shared. However, at the moment, the Darwin Core archive is what we use. And basically, it looks like this. So Darwin Core is that shared community vocabulary, so that's developed with the community and very widely used. And then what we do is in a Darwin Core archive, which is pictured here with these different boxes that represent tables, you would have a core table, and then you have extension tables. And basically, and also the red are then metadata and environmental metadata tables, which these are all combined into a zip archive, and that's what we call the Darwin Core archive. So for eDNA data, what you will see is that you can use an occurrence core or an event core, and then use the DNA derived data extension. And that's just the really basic. So you would have two different tables representing different fields, different column names, basically. That's the vocabulary, is the column names. And those are linked together in a zip archive. We have a guidebook available, which was developed together with GBIF, publishing DNA-derived data through biodiversity data platforms, that has all the details on how this is done, what the extension is meant to do, and how is it meant to be used. So if you would like to look into more detail, that's a great source of information. In this guide, we divide DNA data into 5 different categories. I'm only going to talk about one of these now, and I think in this workshop, we only talk about the metabarcoding data. So that is category 1, DNA-derived occurrences. So that means that species in a time and place, that would be one occurrence. We would also have enriched occurrences, which are then voucher specimens that are barcoded, targeted species detection, which is qPCR, named references, which are sequences in GenBank, and then metadata only, so it would be a data set without sequences. There is a decision tree in the guidebook, which can help you confirm your category if this is unclear. So really, basically, what is the most important, actually the only thing you really need to do is to change your metabarcoding outputs from long format to a short, from a short format to a long format. So basically, when you typically have is several different tables that you get out from a metabarcoding study. So for example, an OTU ASV table, a taxonomy table, where you have your taxonomic IDs linked to each sequence, your metadata, so everything linked to your samples, and of course your FASTA file where you have the actual DNA sequences that are linked to your OTU or ASV IDs. Now, this, I think all eDNA practitioners or DNA practitioners, this is how we usually work with our data. However, in Darwin Core, in OBIS and GBIF, what we do is we work with occurrences. So that means is one row is one unique sequence in one sample. So that means that you're taking your OTU ASV table and you're putting that into a long format. So every ASV in every sample, that is one occurrence. So if we look at what does this actually look like, here you have those 4 different tables that are most commonly used: the taxonomy table, the sample information table, the FASTA file, and the OTU ASV table. And what you basically do is you put all of those ASVs in each sample onto their own row, and you fill in then all the information in one row for that ASV in that sample. So then these become the counts that you have in your OTU ASV table. Here we have just random column names. So basically the next step is then mapping those column names to the correct Darwin Core vocabulary. So the key fields to include in the mapping of the fields that you have in your long format table in the Occurrence Core table basically is the occurrence ID. So that's a stable ID that you will build yourself from the information that you have. Your scientific name, and that links to WORMS, the World Register of Marine Species, and I'll show you soon what we expect you to do with that. Basis of record, so that's always material sample for eDNA samples. Then, of course, your dates and your coordinates, and occurrence status present, which is also always the case. These are the mandatory fields. And then for the strongly recommended fields, we have the scientific name ID, which comes from that WORMS database. Organism quantity, so that's the read count of sequences. That's basically the numbers you have in your ASV table. Associated sequences, a link to those raw sequences and where you have shared them. Organism quantity type, this doesn't change. So this is DNA sequence reads. Sample size value is a total read count in the sample. So by recording this sample size value, what we are trying to do is keep that information on the total amount of reads in your sample so that you can go back to the relative abundance of your reads for that ASV and hopefully get a little bit of an understanding how prevalent it was in your sample. And here as well, you use the sample size unit, which is always the same. And of course, you can have a link to your sampling protocol. So if we look at what is the WORMS, the World Register of Marine Species, and how you get to that scientific name ID, this is a very important step. And you can use several different ways to do that. There is a WORMS taxon match tool, which you can find online and where you can upload your scientific names and it will find you the AFIA IDs. But we also have R and Python packages to do this. WORMS does not have every marine species. If you find a species that is not in WORMS, you can always request to add it to WORMS. Or you can use the highest ranking taxon, the highest rank of the taxon that you do find a match in, for example, family or order, and keep your original information in other fields. In addition, as we know, there will be a lot of unknown sequences, so sequences without species names, and those you can record as biota inserte sedis, which is basically, we say, that is the highest level that you can have, and that is then AFIA ID 12. And then if we look at the DNA extension, so important fields to include is, of course, the actual DNA sequence. your target gene, so what was the targeted genetic region, your PCR primer, both forward and reverse, and this has several fields. So it has the primer actual sequence, the primer names, the reference to that primer, and so on. So you can really in detail record that information so it's easy to understand and search later on. Also sequencing methods, something that comes directly from Mixes. And the other fields for your bioinformatics. So for example, what was your pipeline? What was your database? And the SOP you could use, for example, to record a link to your bioinformatics pipeline that was used for the analysis. In the OBIS manual, you have all this information, and we have also this quick start guide where you can quite easily find the different fields that would be important for each of these different tables that you would record. Now, there is also a different way to do this. So now I've shown you the Occurrence Core and DNA Derived Extension tables, where we link that information via the occurrence ID. But we have a second option where we would use the event core with an occurrence extension and DNA derived extension, both. So in which cases would you like to do this? It adds a tiny little bit of complexity, but this you would use if you have a lot of sample level metadata that you would like to include, for example, on how the samples were collected or different physical or chemical measurements that you took in your event. And if you have that, then it is recorded only once per sampling event. So if you use the occurrence core, you basically record this information for every occurrence. But as we know in DNA datasets, you can have thousands of occurrences, in which case it is easier if you can record these for your event instead of your occurrences. And so what it looks like if you have these three tables is basically in the event core, you will have information on the dates and location and other information that might be linked to your actual sampling. In your occurrence, you then have mostly your taxonomic information and these bases of record and occurrence status. And then, of course, as before, the DNA-derived data extension has your sequence and sequencing information. In addition, you can have a fourth table, which is the extended measurements of facts that I'll also talk about a bit later. And here, this is just depicted what it looks like when you have this organization of tables. So the event core links to your occurrence extension, which links to the DNA-derived data extension. And then you can have the extended measurement or fact, which very flexibly allows you to record your physical and chemical measurements of the sampling event. So the extended measurement or fact extension, it's a table to record all types of measurements. So these can be abiotic, biotic, or environmental. And they are also recorded in a long format. So what you record is the measurement type. So for example, in here we have temperature, measurement value, which is 25, and measurement unit is degrees Celsius. And then you have also linked to here measurement type ID and measurement unit ID. And these are part of a controlled vocabulary where the idea is that you have very machine readable information. So there's no ambiguity in what this term actually means, because people write these measurements in different ways. They have different terms for them. But these controlled vocabularies allow us to know exactly what you are measuring and how. And you can find that in the NERC vocabulary terms. We have a section in the manual explaining this, and we can also explain this more if you would like to look into adding these vocabulary terms, and if you have a lot of measurements that you would like to add to your events. So that's really the basics of the types of tables that you would have and the standards and the standard fields. It's really quite simple. And then what you would actually do to publish that data is you use the integrated publishing toolkits that are hosted by OBIS nodes. And you can find yours on our website. So as I said, you format your data, you build your core table plus extension. Well, you first format to long format, then you do the mapping of the terms. You join an OBIS node. and their IPT account, so you don't need to get your own account. You upload your data and add metadata, or the node does that for you. You can discuss that with them. And then that is validated and published. And there are several tools that can help you do this, as we also hear during this workshop. For example, the GVIF meta barcoding data toolkit has been very popular so far, and I'll also explain that a little bit soon. We have the R package Robus, and the OBIS tools R package, and of course, just to mention again, the OBIS sequence search, which is then in the data access side. So the metabarcoding data toolkit is a tool developed by GBIF specifically for eDNA metabarcoding data. So what it does is it accepts these tables that we are all using from our outputs, and also other common pipeline outputs, for example, from QIIME2 or Data2. And it handles that wide to long transformation, so getting all those tables into that occurrence format. It generates also automatically the Darwin Core archive and DNA extension. So it can be very, very helpful, and also integrates with IPT publishing. It is still under active development. So for example, currently it's not possible to use the event core structure. And it's also not possible to have a mix of different primers in one data set. So there are certain cases where you either then submit several data sets depending on the primers, or you can do it manually in many different ways. So as you can see, it's not a very complicated process, but of course at first it takes a while to wrap your head around it. Here is just a bit different points where we do quality control when data is submitted. So we look at for AFI-ID for that worms match, and this helps keep the taxonomies comparable with each other, both through traditional methods and eDNA methods and other methods. The format of the decimal degrees is defined, and we look that it is actually found in the marine realm and not on land. The format of the date is defined. That is also checked. And then we look for unique and stable occurrence ID and event ID, and those can be built by you based on the data you have in. So for example, can be sample or like date, location, sample, ASV, for example, can be an occurrence ID, and if the table linkages are consistent. There are some tools also for validating the these different quality steps that you can look into. So that's really it in a nutshell. I have just a few more slides, but to look more in detail, you can look at the OBIS manual, the GBIF DNA publishing guide. We had a training in 2024 that has some nice exercises and other workshops by other members of the OBIS community. And of course, you can always send us an email at obis@thehelpdesk@obis.org. And just to mention, we are also developing a data management platform in connection of one of our eDNA projects, where we will have integrated bioinformatics, quality control of taxonomic annotations, and validation of data sets, and as well as publication to the OBIS pipeline. And this will hopefully be also offered to people outside of this project in the coming year or so. And especially, I would say that the taxonomic validation is a very interesting an interesting part of this platform, because it allows to check that your taxonomic annotation matches what we geographically expect from that location. So you could potentially find errors in your annotations because that species is not expected to be found in your location. So just in a quick summary, there's 3 steps to publishing your eDNA data. It's formatting, getting those data into the tables, the Darwin Core table and the DNA derived extension tables in a long format, and mapping those terms. Document your other metadata. So for example, the primers, methods, bioinformatics, license, and so on, and then publish through your OBIS node IPT. And thank you so much for your attention, and I will see you at the workshop.